Hi, we are from Group RNA, and we're going to present Chapter 12, Genome Dynamics. Let's start with, what is immunogenetics? Immunogenetics is defined as a subcategory of genetics, focusing on the genetic basis of the immune reaction. It explores the relationship between the immune system and genetics. Next is about genes for antigen-recognizing molecules. Genes for antigen-recognizing molecules are the genetic sequences that code for proteins that enable the immune system to detect and respond to foreign antigens, such as viruses, bacteria, and other pathogens. They are frequently present in immune cells, such as T cells and B cells. Let's have a look at the three main antigen-recognizing molecules. BCR is a protein complex located on the surface of B cells. The genes belong to Ig gene family. The BCR allows B cells to recognize specific antigens in the body. When BCR binds to its specific antigen, the activated B cell will divide and differentiate into plasma cells, which produce antibodies to neutralize the pathogen. T cell receptor, TCR, located on the surface of T cells, consists of alpha and beta chains in most T cells, or gamma and delta chains in a smaller subset. It allows the recognition of antigens presented by infected cells through the major histocompatibility complex, MHC, activating T cells to perform immune responses like killing infected cells or aiding other immune cells. The major histocompatibility complex, MHC, are highly polymorphic. They are cell surface proteins that present antigen fragments to TCR. They act as antigen presenters, allowing T cells to recognize pathogens. MHC class I presents antigens to CD8+ cytotoxic T cells for killing infected cells, and MHC class II presents antigens to CD4+ helper T cells for activating other immune cells. Now let's talk about the source and nature of genetic variation. Autoimmune diseases are complex genetic conditions caused by defects in the immune system, where the body attacks its own cells causing diseases, such as type 1 diabetes, multiple sclerosis, rheumatoid arthritis, and Crohn's disease. Let's explore the genetic pathways leading to autoimmune diseases. Genetic variability, arising from single nucleotide polymorphisms and larger genetic elements like transposable elements, introduces significant changes by causing insertional mutations or altering gene expression. This can lead to immune dysregulation and an increased risk of exacerbating autoimmune diseases. There are three types of transposable elements, which are insertion sequence elements, transposons, and bacteriophages. So, what are insertion sequence IS elements? IS elements are the simplest form of transposable elements found in bacterial chromosomes and plasmids. They encode only genes required for their mobilization and insertion. Their size ranges from 768 BP to 5 kb. IS1 was identified in E. coli's galactose operon and can exist in 4 to 19 copies within its genome. The ends of all known IS elements contain inverted terminal repeats. The integration process begins with transposase recognizing the ITRs at the ends of the IS element, creating staggered cuts at the target DNA site, inserting the IS element into the host genome, and filling in the gaps using host DNA polymerase and ligase. This resulted in duplicated target site sequences. Next, transposons, also known as transposable elements, are segments of DNA that can move within the genome through a process called transposition. They are often referred to as middle repetitive DNA, which contribute genetic changes, such as spontaneous mutation, genetic rearrangement, and horizontal gene transfer. Let's look at Barbara McClintock and her discovery of jumping genes. The discoveries are AC or DS system in maize. The observations are unstable elements where AC can activate DS causing unstable genetic expression, changing map position where transposition of DS caused altered map positions causing genetic instability, and chromosomal breakage, which DS induces chromosomal breaks causing rearrangements in the maize genome. In the 1970s, Barbara McClintock's research was extended to the rediscovery of bacterial IS in bacteria. These elements function similarly to AC or DS system and contribute to polar mutations. IS cause discrete changes in the physical length of DNA, similar to the effects observed in maize. Which, like AC or DS system in maize, both AC or DS and bacterial IS elements have inverted repeat ends that are crucial for their movement. After denaturation, inverted repeat ends form characteristic lollipops in electron micrographs, providing a clear visual representation of the transposon's mobility and offering insights into the molecular mechanics of genetic rearrangements. What are the key features and aspects of transposons? Transposable elements have a distinct structure that includes a transposase gene, which encodes the enzyme responsible for catalyzing the movement of the transposon, as well as terminal inverted repeats, which are crucial for recognizing the sequence. Next is the mechanism of transposition, involves the excision of a transposon from its original genomic location and its subsequent insertion into a new site, facilitated by the transposase enzyme, which can lead to various genetic alterations such as gene disruptions, duplications, or rearrangements. Lastly, the types of transposons. DNA transposons move as DNA molecules and involve the direct excision and reintegration of the transposon DNA, while RNA transposons are transcribed into RNA and then reverse transcribed back into DNA before integrating into a new genomic location. Let's look at the impact and significance of transposons. First is genomic impact. Transposons contribute to genome evolution by promoting genetic rearrangements, creating mutations, and influencing gene expression. Second is genetic diversity. As transposons move around the genome, they can generate genetic variation, which may be subject to natural selection. Third is the role in evolution, which transposons contribute to genomic innovation and adaptation. Lastly is application in genetic engineering, which the ability of transposons to move and integrate into genomes has been harnessed for genetic engineering purposes. Transposon-based systems have been developed. How many different mechanisms of transpositios? 3. 1. Conservation transposition is the element itself moves from the donor site into the target site. Conservation transposition has a structural feature, which has a transposase gene that encodes an enzyme for catalyzing the movement. The movement mechanism involves the excision of the transposon and insertion into a new location. Lastly, DNA-mediated transposition moves as DNA. The transposase enzyme directly excises the transposon DNA and facilitates integration into a new genomic location. This illustrates the mechanism of conservative transposition. 2. Replicative transpositions show the element moves a copy of itself to a new site via DNA intermediate. Their structure consists of a transposase gene and IRS. They also have transposon-specific resolvase, or recombinase. The transposon is duplicated. The transposase enzyme mediates the transposon's excision, and the transposon-specific resolvase facilitates the integration of transposon copy. The duplication left a copy at the original site, causing an increase in transposon copies. This illustrates replicative transposition. 3. Retrotransposition shows the element makes an RNA copy, which is reverse transcribed into cDNA and reintegration. RNA-mediated transposition uses RNA intermediate for transposition. The retrotransposon is transcribed into RNA. Then the transcribed RNA is reverse transcribed into DNA by enzyme reverse transcriptase. This newly synthesized cDNA is integrated into a new genomic location. There are two types of retrotransposons which are long-terminal repeat, LTR, retrotransposons, and non-LTR retrotransposons. 4 LTR retrotransposons have long terminal repeats at their ends, resembling those found in retroviruses, while for non-LTR retrotransposons lack these repeats. This illustrates the retrotransposition process, which involves key steps such as transcription, reverse transcription, and reintegration. The common features of mobile genetic elements included the generation of short direct repeats, flanking the newly inserted element. This results in a staggered cut being made in the DNA strands at the site of insertion. As the transposon inserts itself, these staggered cuts are filled in, causing direct repeats on either side of the inserted element. Transposons are similar to IS elements, but are more complex structurally and carry additional genes. There are two types of subtypes of conservative transposons. First is composite transposons. Tetracycline resistance can be carried by a composite transposon, which consists of IS elements. IS10R, an autonomous element that contains open reading frames, ORFs, encoding the necessary proteins for transposition, and IS10L, a non-autonomous element that lacks the genes required for reverse transcriptase or transposase. Non-composite transposons are distinct from composite transposons, as they carry additional genes. Their ends consist of non-IS element repeated sequences. For example, TM3 includes 3 key genes, FLA, TMPA, and TMPB. Now we'll move on to third TE, which is bacteriophages. Bacteriophages are defined as viruses that infect bacteria that play roles in bacterial genetics, evolution, and ecology. Bacteriophages consist of capsid that contains their genetic viral genome, tail for attachment to bacterial surfaces, and injection of genetic material. The genetic material of phages exists as either single-stranded or double-stranded. An important feature of phages is their host specificity. Each type of phage targets specific bacterial species or strains. For characteristic of viruses are non-cellular infectious agents that consist of a nucleic acid genome encased in a protective protein coat, and in some cases, additional layers. They are obligate intracellular parasites, meaning they can only replicate inside a host cell. Viruses have several key components. The nucleic acid core contains the viral genome, either DNA or RNA, but not both, and may be single-stranded or double-stranded. It encodes the information required for replication and assembly. Capsid, which is made up of capsimeres. The capsid serves to protect the viral genome from environmental damage. It determines the virus's shape. Lastly, some viruses may contain enzymes for replication within the host. There are some applications of bacteriophages. They are used as an alternative to antibiotics, as vectors in genetic engineering, as regulation of bacterial populations, and to control bacterial contamination in food. Now let's have a look at the virus structures. First is the viral genome. The viral genome is a single or segmented, circular or linear molecule of nucleic acid functioning as the genetic material of the virus. It can be single-stranded or double-stranded DNA or RNA, but almost never both, encodes for the synthesis of viral components and viral enzymes for replication. Next is viral capsid. The capsid or core is a protein shell surrounding the genome and is composed of protein subunits called capsimeres. The capsid serves to protect and introduce the genome into host cells. Some viruses consist of no more than a genome surrounded by a capsid and are called nucleocapsid or nucleocapsid. Attachment proteins project out from the capsid and bind the virus to susceptible host cells. Lastly is viral envelope. Many animal viruses are enveloped which have a lipid bilayer surrounding their nucleocapsid. The envelope is derived from the host cell's membranes, such as the nuclear membrane, Golgi apparatus, or outer cytoplasmic membrane. Virus inserts its proteins and glycoprotein spikes into the envelope. Glycoproteins allow the virus to bind to specific receptors on the surface of host cells. Here are some viral envelope examples such as rubella virus, influenza A virus, HIV, and coronavirus. Now let's learn about the viral replication cycle. Virus replication cycle is the sequence of virus infect host cell, replicate genetic material, produce viral components, and release new virus particles. Firstly is attachment, where viral proteins on the capsid bind to receptors on host cell surface. Secondly is penetration. The attachment process can cause changes in the viral capsid, causing the fusion of the viral and host cell membranes. Thirdly is uncoating. The viral capsid is removed by viral or host enzymes, releasing the viral genome into the host cell. Next is replication. The viral genome is transcribed or translated. Then is assembly. Newly synthesized viral proteins and genomes are packaged into new virions, ready for release. Lastly is virion release. There are two methods of viral release. Lysis is the host cell bursts, releasing the viruses and killing the cell second, while budding is the virus exits the host cell by budding off, acquiring an envelope without killing the host cell. After release, some viral proteins stay in the host cell membrane which can be targeted by antibodies. We'll now move on to virus-host interactions. Virus-host interactions refer to the complex relationship between viruses and their host organisms. Types of virus-host interactions. 1. Productive infection. Virus replicates and produces new virions. 2. Latent infection. Viral genome persists in host without active replication. 3. Chronic infection. Virus replicates at low level without severe symptoms. 4. Transforming infection. Virus alters host cell's normal growth. 5. Abortive infection. Virus enters the host cell, fails to complete replication cycle. Let's discuss about HIST response to viral infection. Innate immune response is the first line of defense, involving interferons, natural killer cells, and macrophages. Adaptive immune response involves T cells and B cells. Cytopathic effects is the structural and functional damage caused by the virus to the host cell. Evasion of host immunity is the virus's evade immune responses through mechanisms. Here are some outcomes of virus hosts such as acute infection, persistent infection, oncogenesis, and host death. Now, let's have a look at the difference between lytic cycle and lysogenic cycle. In the lytic cycle, the virus immediately hijacks the host cell, makes new viruses, and causes the cell to burst, destroying it. In the lysogenic cycle, the virus integrates into the host DNA and stays dormant until triggered to start the lytic cycle later. This picture illustrates both lytic cycle and lysogenic cycle. In lytic cycle, attachment, penetration, uncoding, replication, and synthesis, assembly, and virion release occurred. In lysogenic cycle, bacteriophages undergo attachment, penetration, integration, and replication. When phage is activated, it will enter lytic cycle. That's all from our group. Thank you for watching.